Hello guys, welcome back. So if you saw the previous video, so we are studying about the small organic molecules in the cell, and we went through sugar, fatty acids and amino acids. Now we'll study the last major small organic molecule, that is nucleotides. Right, so I draw a picture. I would post a picture here as well. You will see the image as well, the clear image. So the nucleotides are subunits of DNA and RNA. So the nucleotide, what are nucleotides? So they are subunits of DNA and RNA. So that means DNA and RNA are built from nucleotides. There is a question, nucleotides are nucleosides. So what are nucleosides? Nucleosides are made of nitrogen containing ring compound. So this is the nitrogen containing ring compound linked to a 5-carbon sugar. So this is linked to this 5-carbon sugar, 1, 2, 3, 4 and 5. So this is a 5-carbon sugar. Okay, this is a sugar which can be either ribose or deoxyribose. Alright, nitrogen compounds. So the nucleotides are nucleosides that contain one or more phosphate group attached to the sugar, and they come in two main forms, those containing ribose that are called ribonucleotides, and those containing deoxyribose are known as deoxyribonucleotides. Right, so the nucleotides are nucleosides, but what are nucleosides? So the nucleosides has a nitrogen containing ring compound, the base, then a carbon, 5-carbon sugar, that's this. It can be ribose or deoxyribose. So the nucleotide are nucleosides that contain a phosphate group, one or more phosphate group attached to the sugar. Alright, and they come in two main form. Which main form? Two main form. So one is ribose nucleotide, and the other is deoxyribonucleotide. Alright, so those containing ribose sugar, it will be known as ribonucleotide. Those containing deoxyribose sugar are known as deoxyribonucleotide. Right guys, so that was a definition or an introduction for the nucleotides. So the nitrogen containing ring compound, this ring compound of all these molecules are generally referred to as a base. And why they are referred to as a base? Because of a historical reason. Under acidic condition, they can easily attach to an hydrogen H+, that is hydrogen atom or proton, and thereby increase the concentration of hydroxyl ion in aqueous solution. So that means it can lower the acidic solution and make it alkaline. So that's why it is known as base. Alright, because it can attach to an H+, the proton, and thereby increase the hydroxyl ion concentration in an aqueous solution. So there is a strong a strong family resemblance between the different nucleotide bases. So there is a strong family resemblance between the different nucleotide base. So there are different type of nucleotide bases. So they are cytosine, thymine and uracil, that are called pyrimidines, because they all derive from a 6-membered pyrimidine ring. That is why they are called. So I'll write it down here, maybe. So cytosine, 1, thymine, and the third is uracil. Alright, so these are called pyrimidines. So put this here. Pyrimidines. And why they are called pyrimidines? Because they all are derived from a 6-member pyrimidine ring. Alright, there is guanine and adenine are purines as well. There are two different kinds as well. So they are in, and the fifth is adenine. And this are called purines. Alright, so which bear a second 5-member ring fused to the 6-member ring. Alright, so the difference is why it's pyrimidine, because they have a 6-membered pyrimidine ring, and purines, which has a second 5-member ring fused to the 6-member ring. So each nucleotide is named after the base it contains. I am posting a picture. You will see this image. So that is showing the nucleotides and the abbreviation, and it shows on the side. So the base. So this base. So in every nucleotide, so you know there is this ring compound, nitrogen ring compound, sugar, and the phosphate group, and this is the base, and the base changes. So that is adenine. So the nucleoside is adenosine. Guanine, the nucleoside is guanosine. Cytosine, the nucleoside is cytidine. Then the uracil, it's uridine, and thymine is thymidine. Alright, so that is the nucleoside, as you can see in your screen, and it has the abbreviation A, G, C, U, and T. Right, so AMP means adenosine monophosphate, DAMP means deoxyadenosine monophosphate, UDP means uridine diphosphate, and ADP means adenosine triphosphate. Right, so as on the picture as well, you can see the base plus sugar is equal to nucleoside, but base plus sugar plus phosphate is a nucleotide. Alright, so nucleotides can act as an short-term carriers of chemical energy. So one of the function of nucleotide is it can act as a short-term carrier of chemical energy. Carriers of chemical energy above all other. So if we exclude the AMP and DAMP, the ribonucleotide adenosine triphosphate, or ATP, participates in the transfer of energy in hundreds of metabolic reactions. So in so many metabolic reactions, the ATP always takes a part in the transfer of energy. So the ATP is formed through reactions that are driven by the energy released by the breakdown of foodstuff. And so its 3 phosphates are linked in a series by a phosphoanhydrate bonds. So the bond that is joining the 3 phosphate is, so you can see on your screen, like I posted a picture of that. So that has this and 3 phosphates with the phosphoanhydrate bond. So that is an ATP. So a rupture of this phosphate bonds releases large amount of useful energy. So when this phosphate bond is broken down, it releases large amount of useful energy. So the terminal phosphate group in particular is frequently split off by hydrolysis. So the last one, so if there is 1, 2, and 3, and the last one is split usually off by hydrolysis. So that means the water is added to the reaction. So in many situations, transfer of this phosphate to other molecules release energy that drives energy requiring biosynthetic reactions. And other nucleotide derivatives serves as carriers for the transfer of other chemical groups. We'll discuss later on when we'll start discussing about the DNA, RNA in more details. So the nucleotides also have a fundamental role in the storage and retrieval of biological information. So apart from the transfer of energy, that's one of the function of nucleotides. The main fundamental role of nucleotide is storage and retrieval of biological information. So this serves as a building block for the construction of nucleic acid, long polymers in which nucleotide subunits are linked by the formation of covalent phosphodiester bonds. Right, so the nucleotide serves as the building blocks for the construction of nucleic acid, long polymers in which nucleotide subunits are linked by the formation of covalent phosphodiester bonds between the phosphate group attached to the sugar of one nucleotide and the hydroxyl group on the sugar of the next nucleotide. Right, so in the image on your screen you will see, so that's a short length of one chain of deoxy, that's a DNA molecule showing the covalent phosphodiester bond linking 4 consecutive nucleotides. So this is how the 5' has a phosphate group, and the third 3' has a hydroxyl group. This it's just say, oh, but it's like this. So the last end has an hydroxyl group. So like the bonds link specific carbon atoms in the sugar ring known as 5 and 3 atoms. One end of a polynucleotide chain, the 5 end has a free phosphate group, as you can see in the image, and the other end 3 has a free hydroxyl group, and one of the nucleotide thymine, you can see that's in the gray, and the phosphodiester you can see that's in yellow. Right, so the linear sequence of nucleotide in the polynucleotide chain is commonly abbreviated by one letter code. We'll study that later. So like you can see, it's GATC, it's the sequence of the letter, and the sequence is always read from the 5. And so the nucleic acid chains are synthesized from energy-rich nucleoside triphosphates. So the nucleoside triphosphate by a condensation reaction that releases inorganic pyrophosphate during the phosphodiester bond formation. So how this are linked to the another one, so this is one, and then it will join to another subunit. So what kind of reaction? It's the same condensation reaction that releases inorganic pyrophosphate during the phosphodiester bond formation. So there are two main types of nucleic acid which differ in type of sugar contained in their sugar phosphate backbone. So it's just different. There is two types of nucleic acid that's really famous. One is RNA. Why it's RNA? Ribonucleic acid, because the sugar is ribose, and contain the bases. What kind of base? It contains the RNA. So it has A, adenine, G, guanine, C, cytosine, and U, uracil. So this 4 bases are with the RNA. Those based on DNA, that's deoxyribose. All right, nucleoside. So that means the sugar is deoxyribose. All right, and contains the bases. What kind of bases DNA contain? A, that's adenine, G, guanine, C, cytosine, and instead of uracil, it contains T, thymine. Right, so T is chemically similar to the uracil in RNA. So RNA usually occurs in cell in the form of a single-stranded polynucleotide chain. So in the cell, the RNA is always a single-stranded polynucleotide chain, while the DNA, it's a double helix. That is 2 polynucleotide chain. So the linear sequence of nucleotide in a DNA or an RNA molecule encodes genetic information. The 2 nucleic acids, however, have different roles in the cell. DNA with its more stable hydrogen bonded helices act as a long-term repository for hereditary information, while single-stranded RNA is usually a more transient carrier of molecular instruction. So what RNA does? DNA stores the information, but the RNA is like taking a molecular instruction. It's like an instructor. It will take an instruction from the proteins and send to the other part for how the molecules should be arranged and everything like that. The ability of the bases in different nucleic acid molecule to recognize and pair with each other by a hydrogen bonding, that's called base pairing. G with C, A with either T or U underlines all the hereditary evaluation, and this we'll study later when we'll discuss in detail with the DNA and RNA. Alright, but that's the ability of base in different nucleic acid molecules to recognize the pair with each other by the hydrogen bonding. That's called the base pairing as well. It was nucleotides. So we saw the functions and what are nucleotides. Alright guys, so that was nucleotides. We'll study more in details when we'll discuss about DNA and RNA in a detailed topic. Alright guys, so we'll see you in the next video with the next topic for the cell. We'll keep continuing the cell and we'll move through the system as well after we finish the cell and the basics of it. Alright, thank you guys for watching.